All right, everyone, welcome to chemistry final review. This won't be as long as the first one because this is a comprehensive review. So to review the first half of the course, I suggest going to week 5 and clicking on modules, of course, and then going down to the bottom, and you'll see that. So I'm only going to cover the second half of the course here. So if you want the whole package, just go back to week 5 and look at the exam review, and then come back and do this one for the complete exam review. So the way that the exam is formatted is quite simple. It's half and half. Makes sense, right? So 25 questions will be weeks 1 through 4, and then of course 5 through 8 will be questions 26 through 50. So there are 50 questions total on the exam. So the first couple of questions, I'm actually going to turn my paper this way. First couple of questions that I ask would be in regards to how many carbon atoms, this thing has. So we're going to write out that funny little acronym that, there it is. So remember this acronym, well, it's not an acronym. It's just a funny mnemonic device, right? So we had Molly eats peanuts because peanuts help her ovulate, right? It's really funny, and it's just really funny. So Molly eats peanuts because peanuts help her ovulate. If you remember this, you should be good to go. So what does this mean? Well, this means that anything that's starting with an M has 1 carbon. Anything with an E or an A has 2 carbons. P is 3 and 4 and 5 and 6 and 7 and 8. Now, where students tend to get mixed up is these two right here, right? H and H. Well, this would be hex and this would be hept. So an example question that I might ask regarding this is how many carbons are in methane? Well, it begins with the letter M. So M, therefore it has 1 carbon. How about hexane? Well, it's hex, and here's hex here. That must be 6 carbon. What about ethanol? Well, it begins with an E, so we go to E, and there's 2 carbon. So hopefully that clears that up. And that's going to be a couple of questions on the exam in regards to how many carbons are in what. And that's just the best way. So remember, it's Molly eats peanuts because peanuts help her ovulate. And then again, the other thing you have to remember is the difference between these 2 H's, hex and hept. So yeah, that's the only thing you have to just really memorize there. And again, you're just taking the first letter and matching it, and you should be good to go from there. Let's see, what else? Let's see, that takes care of 1, 2, 3, 4 questions right there. Just double checking here. Okay, and a lot of this class is based off of monomers and polymers, right? So monomers, spell like this, and polymers. So just to give you a few examples of each. A monomer is a building block, right? Because mono means 1, poly means many, and this is the final product. So monomers, in this case, let's say, let's take the polymer first. So a polymer here would be lipids. All right, so if the polymer is lipid, I want to know what makes up a lipid. What's the monomer of a lipid? Well, it would be fatty acids. Fatty acids make up lipids. Well, what about another one? This is a little ambiguous, but We'll say carbs or carbohydrates, of course. Well, the monomer of carbs would just be sugars. All right? Protein. What makes up a protein? Amino acid. And so on. So those are your polymers. Those are the big, these are the macro. We'll call them macros. Because these are the big elements here. Lipids, carbs, and proteins, those are the big ones. But the monomers are the ones that make up the lipids, that make up the carbs, and that make up the protein. And there's other examples too, but these are the big three that I want you to know. So you need to know what the difference between polymer and a monomer, and then you need to know all of what makes up lipids, carbs, proteins, nucleic acids, and so on. And that you'll just find, in your studying. And again, these prefixes here, poly and mono, are going to be very important because mono means one, poly means three or more. So what's in between these two? Well, we sometimes call them di, like a disaccharide, but we really don't use that too often other than just sugars, of course. This is a good example. The polymer is DNA. What's the monomer? Well, the monomer would be nucleic acids. And that's what the Na in DNA stands for, right? Deoxyribonucleic acid. So know your polymers, know your monomers, and then, of course, as I mentioned before, know how many carbons are found in various molecules here. Now, in addition to this, I'll get rid of this paper. In addition to this, you're also going to want to know hold on a second here. No. So on this exam, let me just double check here. Yeah, you're going to want to know, carboxylic acids, esters, and ethers, and all that fun stuff. But that's just simple memorization. So go back to the PowerPoints and memorize it, but don't overdo it. I think there might be one question on the exam about that, so don't overdo that one. But knowing how many carbons that's a few questions right there. Let's see. Let's talk a little bit more about DNA. So sorry, I'm just trying to figure out where to start here. Let's start with DNA replication, transcription, translation. So with DNA replication, I'll just put DNA rep here on the top. We start off with DNA. I'm just going to draw a little squiggly line and then another squiggly line that sort of intersects that. It's supposed to be a double helix. With DNA replication, we want to make more DNA, right? That makes sense because that's what replication means. Well, what do we need to do this? What enzyme is necessary? We need an enzyme called DNA polymerase. Because remember, in the lecture that I had recorded, DNA polymerase makes DNA, and RNA polymerase makes RNA. So we need DNA polymerase. And what that's going to do is that is going to create two different strands here. Oh, sorry. Again, I know my drawing's terrible. It's okay. These are two different DNA molecules now. At least that's what they're supposed to be. So there we go. So now we have one DNA molecule and we have another. And for the focuses of this class, in terms of replication, that's all you need to know. But what about base pairing? Because DNA, as we saw, I'm going to flip it over. DNA, as we saw, is a double helix, right? So if we go through here, this, you'll see all of these bases on the inside. Now, what bonds to what? Well, if it's A in DNA, I'm right there here on top, DNA. If it's A, it bonds to T. If it's G, it bonds to C. And if it's T, it bonds to A. And there's one more. If it's C, it'll bond to G. Those are the base pairs. So let me write it out plain English for you. A to T. There's the 2A arrow here to indicate that T also goes to A. And then C, double arrow, G. So, and this is for DNA only. Because the rules change once we go there, right? So we had DNA replication, which is a horrible representation of that, but still. DNA replication, we go from one single DNA to two DNAs using DNA polymerase. And then the base pairing goes like this, right? And it's up here. Now the whole point, or what we call the central dogma of biology, is to go from DNA to a protein, because proteins make up just about everything in the body. So the question now becomes, how do we get there? How do we go from DNA to protein? Because DNA, it's inside the nucleus, it doesn't leave. It can't make protein on its own, but it can elicit some help. And so now we're going to talk about transcription and translation. So we'll talk about transcription first, because that's the first step anyway. So I'm going to draw a nucleus on the corner of my paper here, and I'm going to draw just a little squiggly line to indicate that that's DNA, right? So transcription, always happens in the nucleus. Always happens in the nucleus. If it's happening outside the nucleus, it's translation. So let's kind of zoom in on this to see exactly what's going on here. So we have, again, large DNA molecule. And we have all of our bases. And now I want to make mRNA. Now, if I want to make RNA, what enzyme do I use? Hopefully you're at home screaming at your computer saying, yeah, you use RNA polymerase. RNA polymerase, just like the last one. RNA polymerase. Well, what is the RNA polymerase doing? Well, it's making one strand of DNA. I'm sorry, one strand of RNA, excuse me. So what it's doing is it's splitting up DNA, and now it's going to make its own strand. But let's take some examples here. So let's say we have an A, we have a C, we have a G, and so on. That's just one codon right there. Well, because this is mRNA, since this is RNA, we now have to use different sets of rules. So before we figure this out, let's talk about the rules. So in RNA, there is no T at all, right? So instead, A bonds to U and U bonds to A, but C and G are still the same. So A bonds to U, U bonds to A, C to G, G to C. So with that knowledge, this is the RNA strand. A bonds to U, C bonds to G, G bonds to C. And so what you'll get here is a single strand of all of these different bases, and we know this as mRNA. So I'm going to go back into our zoomed out picture here, and we have one strand of mRNA. So a couple of things have to happen to it first, right? First of all, we have to add a cap and a tail. So there you go, there's our cap and tail. And we don't want all of it. So whatever we don't want stays inside. So I'll just put a couple little fragments there. And we know these as introns because they stay in the nucleus. This, however, is known as an exon because it exits the nucleus. So it's going to come out here. Now that's the whole process of transcription. So we're going from DNA to an mRNA strand via RNA polymerase and using this bonding technique down here. That's it. That's what transcription is, going from DNA to mRNA using RNA polymerase. And then, of course, that would also include cutting out your introns, splicing together your exons. That's it. As I mentioned before, if it's happening in the nucleus, it's transcription. Automatically done, that's it. If it's happening outside the nucleus, it's translation, which we're going to talk about right now. So translation happens outside the nucleus, but I'm still going to draw the nucleus there for reference. DNA is still in the nucleus, hanging out, doing its job. It's fine. It's still DNA. But you notice how DNA never leaves the nucleus. It gets everyone else to do its dirty work for it. But now we have, I'm going to draw this a little bit bigger now. We have an mRNA strand. So I'm going to write that down here. mRNA. And we got 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and so on. Now, these little tick marks, those are nitrogenous bases. They're known as a codon. And a codon is 3 nitrogenous bases. So this could be AAA, this could be CGC, could be UUA, this could be CGG, and so on. I'll just do the rest myself here. So those are all codons. And no, they're not spread apart that easily. They're not. So this is now in the cytoplasm of the cell. That's just the outside of the DNA. And it's swimming around, and it's going to encounter a ribosome. And remember, ribosomes come in 2 parts. So this is a ribosome. And it is made out of rRNA. That's what the R stands for. It stands for ribosome. Well, if you learned in biology class, you know that ribosomes make proteins, but how? How do they make proteins? Well, you're going to get this guy. We're going to draw him as a capital T. And he's going to have a little circle on him. And this guy is known as tRNA. And that T stands for transfer RNA. Well, what is it transferring? It's transferring this guy, and he's an amino acid. Transferring an amino acid. Now, on the bottom of each of these, you'll have this, and that's called an anticodon right there. And I'd label it, but I don't want to, no, it's fine, I'll do it anyway. Anticodon. So that's the anticodon on the tRNA. Let's take a closer look at exactly what happens when they bond together. So let's draw it like this. What should be the anticodon for AAA? Well, I'm going to put the letters on top because I don't think I have room to write them. A bonds to what? Let's go back to our rules here. A bonds to U. So this one must be U U, U. Okay, that was easy. And this is this is the anticodon, and this is the codon. What about UUU? Well, what's the anticodon for that? That would be probably AAA. Not probably, it is. So every time you bring an amino acid down, it bonds to its proper anticodon, you're going to start bringing more amino acids down. And when you start doing that, you're going to start making more and more and more amino acids. And this is going to make a protein, right? So if we kind of bring this guy down here, and just draw a bunch of circles together. And we call this a protein. Now, is it complete? No, it's not. This is a primary structure of protein, but this is protein nonetheless. So just to review, we started inside the nucleus. Remember, transcription always happens inside. I just expanded the view so you can see. We went from DNA to mRNA using RNA polymerase. We cut out what we didn't want and kept what we did. Introns stayed in the nucleus, exons exited the nucleus. Once it exited the nucleus, it came out of here, then this mRNA strand had codons on it. Well, it had codons before it left the nucleus, but still, codons on it. Hooked up with this big old ribosome that comes in two parts, and it is made out of rRNA. And once it did that, you're going to have this tRNA come along and with an anticodon bond to the correct codon sequence and bring that amino acid down. And this happens so many times that you have different amino acids that get bonded together via peptide bonds. And then you made a protein. So we went from DNA to protein via RNA. Now that's confusing. I highly recommend you look up a video on YouTube. It is called, I think it's just called DNA to Protein 3D. It's got like 7 million views on it. It's perfect. I highly recommend you use it. Okay. So that's DNA transcription translation, which does take up a lot of your part of your exams. So make sure to study it thoroughly. I will ask you to bond a couple of things. Like I'll ask you to bond. I'll ask you to transcribe and translate. So, let me get a separate sheet of paper out for that. If I ask you to transcribe, that means we're going from DNA to mRNA, right? If I ask you to translate, that means we're going from mRNA to tRNA, which is just codon to anticodon. Okay. So that's really important to know. And I'll give you a couple of brief examples. I'm just going to do one codon for each. So let's say the codon for DNA is TAC. Okay. The bonding, the transcription for that would be T bonds to A bonds, oh shoot, sorry. I was one step ahead of myself. Apologize for that. TAC. T bonds to A bonds to U, and G bonds to C. So that would be an example of transcribing. Let's take translation. So maybe it's UAC. All right, so UAC, U bonds to A bonds to U, C bonds to G. And that would be translating. By the way, this is a start codon. So anyways, that's how you do that. And there's one final thing that I'd like you to know. Pardon me for a sec. Okay. I don't ask too many questions about metabolism, but I would like to go over it and just go over the basics. So I'm only going to use one piece of paper for this. So metabolism happens in three parts, mainly. It's glycolysis, or glycolysis, but I like to say glycolysis because it describes what it does. Krebs, and then I'll write the whole name: electron transport chain. Okay, so what happens in each one? So in glycolysis requires no oxygen. That's really important that you know that. We get 2 ATP, and we get also 2 NADH and 2 pyruvic. I might be spelling this wrong, so pyruvic acid. It's actually the proper one. The Krebs cycle, we get 6 CO2s. We start out with acetyl coenzyme A, it's capital A, and we make 2 ATPs. The electron transport chain makes about 32 ATPs, plus or minus a couple. Yeah, sorry, it makes 32 ATPs, plus, we'll just say plus or minus 4. It's about right. Your book will say, I think 32 or 28, I forget, but it just depends on who you talk to. It does require oxygen, needs oxygen, so I'm just going to write needs O2. And as far as location, outside the mitochondria, we'll just say outside and inside. Okay. Sorry, we make 6 NADHs and 2 FADH2s here. So essentially what happens is outside the mitochondria, you actually pay 2 ATP to get in, but you get 4 in return, so that's a net 2 of ATP. NAD turns into NADH, and then you make 2 pyruvate from there. And you would then, the 2 pyruvate, that's what goes to the Krebs cycle to be turned into acetyl coenzyme A, and that costs carbon dioxide. In the process of turning the Krebs cycle, we make 2 ATPs, 6 NADHs, FADH2s, and so on. Now, if you didn't watch the lecture, then this wouldn't make sense, but the final step is going from NADHs back to NAD, and same thing with FADH2 to FAD. So basically, in the electron transport chain, we get rid of all of these hydrogen atoms, and all of those hydrogen atoms are going to be, rather electrons, are going to be pumped into the inner workings of the membrane in the mitochondria, and they're finally going to go through an enzyme we call ATP synthase. And that makes the majority of your ATP right there. So yes, it is important that you know those details. However, if you're banging your head against the wall trying to remember all of this in order, I highly recommend just knowing the information that I outlined here, and don't go into too much detail because that's really more or less outlined in biology as well as A&P2. But chemically, that's what I, of course, want you to know. And what are the products, right? What are the products of cellular respiration? Well, the products would be water and carbon dioxide. All right, so I'm going to put H2O here on the bottom because it does make H2O. Remember, oxygen is the final electron acceptor, so that's going to make H2O. Now, what do we need to start this? Well, glyco refers to sugar, so sugar would be C6H12O6. And what do we breathe in? We breathe in oxygen. So this is known as the cellular respiration equation. So we take in oxygen, we eat food that has sugar in it, and then we breathe out CO2 and sometimes water. We are excrete water. Now that's for us, right? Now plants have a different agenda, right? They're going to do the opposite. Plants need carbon dioxide and they need water, right? Because you need to water your plants. They give us oxygen and sugar. So what do you notice about these two equations here? Well, hopefully you know that they're opposite each other. One feeds the other. The products of one equation are the reactants of the other. And this is just known as, here, I'll put plants here. This is the circle of life right here. I mean, this is how we as a species and plants as a species can survive. Now, when I say us, not just human beings, I hope you know that. It's literally every animal, or rather every cell that's not a plant that can do this. So all bacteria, fungi, I should say most bacteria and fungi do this. Well, all plants do this, and they produce a lot of this. Now, there are some algae that can do this as well, and I shouldn't discredit them because they certainly do produce a lot. But anyways, point is, this process feeds that, and then vice versa. And you need to know that because it's super, super duper important. But let me go see if I got everything. Yeah, I got everything. Okay, so first of all, sorry for the dirty fingernails. I was working on something before a couple hours ago, and I washed them pretty good. I thought I washed them pretty good, but then looking underneath the camera, I realized that was awful. Just a couple of reminders. I'm going to put this on paper so we can see it. Labster. You need to make sure all of your labsters are complete by Sunday, this coming Sunday, or you'll get zeros. You don't want zeros. Your lab practical is an average of labster. So if you get a 0 on a labster assignment, it'll be averaged into your lab practical. I know this doesn't say August, it says average, AVG, average of labs. Weekly quizzes. If you haven't already, turn those in, because otherwise you'll get a 0 and that'll bring down your grade and you'll be really sad. So don't do that. So complete your labs, complete your quizzes, and complete your labs so that I can do this. Now, I am responsible for this. I will be averaging your lab practical grades on Sunday, just so I give you as much time as I can. So if you have any questions, do not contact me on Sunday, or Sunday night, I should say. Do not wait to the last minute to do this, because if you do, then I can't guarantee that I can help you. But final exam will be on this coming week. It opens at 6 p.m., closes at, I think I closed it at 10 p.m. It's an hour and a half long. and you'll receive all those details soon. So anyways, if you guys have any questions, please let me know. But again, don't wait to the last minute. I want to help you as best I can, and I need time to do that. So make sure you reach out to me sooner rather than later. So if you have any questions, let me know. If not, good luck. And it's been a pleasure teaching you all this semester, and I really hope you enjoyed chemistry. So take care, guys. Bye-bye.